Hello everyone, and thank you so much for joining me for today's class. Today we are going to look into statistical concepts in clinical research. So first, allow me to go over the drug development process for a few minutes. We always start with a compound that is biologically active. The compound may be from plant, from insects. It may be synthetic, and again it may be also coming from different organisms as well. And we are going to call that phase the discovery phase. So in any pharmaceutical company, we do have a department that is called R&D, or research and development. So what those departments they do is that they are going to find or to use or to work on different I would say products that are not yet pharmaceuticals, but they do have they are biologically active. And we are going now to look into those compounds. We are going to make them stable, maybe trying to again have them in more stable different formulations as well. And then we are going to, of course, looking into how they are going to be used. So we start by looking into the effect of those compounds in vitro. In vitro means that we are going to have petri dishes, for example. We are going to have cell cultures, again bacterial cultures, and we are going to expose those cultured cells or bacteria or any other organism to our compounds, and we're going to see what the compounds are going to do. We may look into just the effect just visually. We may also look into intracellular pathways by doing what we call fundamental research, and we are going to see exactly what those compounds are doing with the target cells that we are having in vitro. So once we have characterized the effect of those biologically active compounds, we are going now to start testing them in vivo or in live organisms. So here we are going to have several animal species, depending on again what is the best animal model or what are the best animal models. We are going to test those products, and once we have characterized the effect of those compounds or the compound in vivo in animals, we are going to look into testing those in humans. This is why we talk about clinical trials. Clinical trials because we are going to have a trial. This is a type of intervention that we are going to to perform using the compound, and we are looking into its efficacy and its safety profile. But again, checking the efficacy and safety profile of a compound did start already when we started in vitro, because we are getting information about what are the intracellular pathways, for example, that are being affected by the effect of the compound. And even we can start in vitro by looking into what are the doses. So we cannot just use one dose of the drug. We may use 10 different doses of the drug, increasing doses, and then we are also going to measure and to give metrics, I would say, into the effect of the drug. And from where we start even to look into doses. So coming back to the clinical trials, we are going to go over several phases in clinical trials. We have 4 phases. Phase 1 is when we are going to test the drug for its pharmacology. We would like to know what the drug does to the body. We call that pharmacodynamics. We also want to know what the body does to the drug. We call that pharmacokinetics. We also want to know what is the influence of genes to the extent of the influence of the genes on the effect of the drug on the body. We call that pharmacogenomics. And then when we are going to look into phase 1 trials, we are exposing healthy populations And of course, sometimes if it's not possible because we are dealing with cancer drugs, for example, and we are not going to expose healthy patients to a cancer therapy, so in this case, in phase 1, we may have also the people, I would say, subjects with already having a disease. And then we are going to, in phase 1, look into the pharmacology, and we are going to look into the safety profile. It means that we are going to expose our subjects in phase 1 to different doses of the compound, and we are going to look into the side effects, or I would say adverse drug reactions, that is defined as any untoward medical occurrence experienced by our subjects. And we are going then to look into what is the safe dose or the dose range that we can use for our drug, future drug, so that it's not going to harm any individual. So at the end of phase 1, we already have the good idea, again, I would say well-established idea about the safe dose of the compound that we are then going to use in phase 2. In phase 2, we are going to have about 100, 200 patients, human subjects again. We talk about clinical trials, and we are going to test the compound within the safe dose in patients who have a disease condition. And then we are going to look into whether or not we have the efficacy of the drug that will be established, in addition to re-establishing, or I would say even reconfirming, the safety of the drug using the specific doses that we chose in phase 1. So in phase 2, we are now looking into whether or not the drug, which is safe, using the safe dose, is effective. And then once we will establish the fact that the drug, the compound, has the efficacy for a specific disease, we will go to phase 3, where we need to have a higher number of subjects, because we need to prove statistically that the effect of the drug that we observed in a very low number of patients is not an accident, and that in fact, based on the statistics, we do have a drug that is working. This is why we cannot just have a low sample size of 100 or 200 people or patients in phase 2. We need to have more than 1,800, 2,000 patients in phase 3. This is why you will have not only one clinical trial, but several clinical trials involving several of hundreds, I would say minimum 1,600, 800 patients, I would say 2,000 minimum, in order to have the statistical power, in order to establish the fact that the effect of the compound that we saw was not an accident, but that was, again, a fact. Then once we are done with phase 3 and we have enough statistical proof that again the drug or the compound is effective and is safe, we are going to continue to prepare our new drug submission for Health Canada, or if we are in the US, our new drug application. If we are in Europe, new marketing authorization application, or in other countries, we're going to prepare those registration application. And then here, as you see, we submit our new drug submission, if you're in Canada, to Health Canada. And once Health Canada will have approved the new drug submission that we submitted based on the clinical trials, based on the quality information, and based on the safety information that we observed and we obtained from phase 1, 2, and 3, and even before in vitro and in vivo studies, we will get our notice of compliance, or NOC, from Health Canada. That NOC, or notice of compliance, is going to allow our company to start selling the compound that is not anymore a compound, because we showed that this is a pharmaceutical product. So our product, our drug, which is now registered, will be again sold on the market. And here we talk about post marketing phase. So post-marketing means when our drug is in the market and we are going to continue to produce the drug, continuing to do some in vitro studies, some in vivo studies, maybe additional clinical trials, because we would like to know more about the product itself, about its safety profile, its efficacy, maybe getting to know more about other indications so we can look into new indications of the drug. So it needs again clinical trials, and we start with phase 2 and then phase 3 because that's a new indication. So anyways, what I'm saying is that when a drug is on the market, it's not the end of the research. We continue to do research, in vitro, in vivo, and clinical trials, because we may want to test our drug in new populations. Maybe we are going to have new formulations. Maybe we have new indications. So phase 4 clinical trials are mainly for the drugs that are on the market, and we are looking into their safety profile as well. So we are going to do studies involving humans, looking into different outcomes. One of them is efficacy. We are still testing the efficacy of the drug in more and more populations, but at the same time we are also going to do studies like pharmacoepidemiological studies as well, where we are going to look into the safety profile of our drug when on the market. So you may see even clinical trials taking place 10 years after a drug is on the market, 20 years after. The best example is aspirin. It has been on the market for more than 80 years now, but recently, 2-3 years ago, we also looked into another indication of aspirin that was for extended release tablets, and that was a new indication. That was for people who were, again, experiencing, again, I would say, a pre-heart attack episode, I would say, and then those people, they have to just crush 2 tablets of extended release aspirin from Bayer, and then they take those 2, they crush them, and then they go to their doctor. This is it. So it's not a treatment, this is a prevention, but this is a preventive treatment for which Bayer obtained again a notice of compliance for a new indication of aspirin that has been for decades on the market. So it's a very, very interesting case as well. And now we understand that when we talk about clinical, we talk about studies involving humans, and we start again those studies in phase 1 through 2, 3. Then on the market, we're going to continue with phase 4. If we are having, again, new indications that we would like to test the drug for, in this case, we have to go back in phase 2 and then to continue with phase 3, creating a new drug submission for the new indication, and then we will have your drug on the market for 2 different indications. So now let's go over clinical research and data. As I already explained, clinical trials, they use humans as participants. The goal of the clinical trials are the following. First one is to ensure patient safety. So in each and every study, we do have questionnaires. We are monitoring patients, and we are going to ask them questions about any untoward medical occurrence that may have experienced during I would say the time when they have been exposed to the experimental drug, and we are going to monitor that. So if there is any issue, we have to, of course, let all of the sites know about those adverse drug reactions that are serious and unexpected. We also need to let all of the countries in which we are testing our drug, again, that yes, we may have some suspected, unexpected, serious adverse drug reactions. And again, it's not going to stop us from continuing to do the clinical trials, unless, of course, the drug shows to be harmful to patients. But usually, again, this is very well controlled. And again, our first job is to ensure the patient's safety. The second one is to determine and understand the effect and value of an intervention in human beings. So we would like to know if our drug, which is safe and we monitor its safety profile, is also having the efficacy that we are expecting in order to treat disease conditions. And of course, to find a better and or new way to treat, cure, and improve health outcomes. It means that many of our drugs may not be healing, okay, but they are improving. It means that, for example, for some types of cancer therapies, we give several drugs. Some of the drugs are for the cancer itself, some of the drugs are for relieving pain. These are analgesics. But again, these are, drugs that have been tested in cancer patients, and we could establish very well their effect, a specific effect in cancer patients as well. So we talk about improving the health outcomes, and I would like to also to ask you to refer to, again, the fundamentals of medicine as well, where we talk about treating, curing, and improving health outcomes. So typically there are 4 phases of clinical studies. I already explained those in the previous slide. We have phase 1, 2, and 3 that take place again during the clinical trial phase. We are not on the market, so we call the drug an experimental drug. When we get our notice of compliance from Health Canada, if you're in Canada, and we go to the market, we start to sell our drug on the market. Then if we do any study for within the approved indication of the drug, but which is on the market, we are going to call that study phase 4. And in phase 4 studies, we may look into, again, the efficacy of the drug in different populations. It may be, for example, in elderly population this time, maybe in pediatric population, because in the clinical, trial phase, before going to the market, we just tested the drug in adult population. So it's one. We may also look into studies where we are going to look into the drug safety profile. And this is part of what we call pharmacovigilance. It means the science of collecting, assessing, reporting, and preventing adverse drug reactions. And through the pharmacovigilance activities, we also have clinical trials that we are looking into the safety profile of the drug, just to discover if there is any adverse drug reactions that we didn't have or we didn't see during the clinical trials, if there is any other drug-drug interactions, for example, that we couldn't see because we were in clinical trials. So we need to constantly monitoring the safety profile of our drug. That's why we have phase 4 clinical trials. So continuing with clinical research and data, the most important factors to consider are patient safety and the questions being asked. I would say data quality as well. But what is the clinical question? So each clinical trial must have a primary question, or we call that a primary outcome in the protocol. So the primary as well as secondary questions that may be related to safety profile must be carefully selected, clearly defined, and stated in advance in the protocol. So the primary question is the one question that the investigators are most interested in answering, and it must be capable of being adequately answered. So the sample size of the patients that we're going to have in a trial in answering, and it must be capable of being adequately answered. The sample size is based on the primary question. It can be framed to test a hypothesis with its postulated outcome. So the outcome may be beneficial or not. It may be looking into drug efficacy or saving a life. You may say that they're all beneficial, obviously, but the outcome is usually something practical. We would like to know the effect of the drug. But you may have in a study not one, not two, but several outcomes. We may look into the effect of the drug on the cardiovascular system, looking into the effect of the drug on the central nervous system as a second primary endpoint. Third primary endpoint will be looking into the safety profile of the drug on overall body systems, for example. So this is how we are going to look into different outcomes. And when we write a protocol, we are going to look into variables that we are going to choose to observe in order to understand more about the efficacy profile or safety profile of a drug. So now let's go over the clinical studies types. So here we have observational and experimental studies. Observational means that we just observe, and experimental means that we have some intervention. So when we observe, we are going to have descriptive types. It means we just describe. We may not do the analysis or explain more. We just describe what we can see. But we can also have, in observational studies, analytical studies. So here we are going to explain. So if you would like to go back to the observational studies and then to go over the descriptive category, we can talk about case reports where we have a patient, it's just one case, we talk about the case, we say what happened, what symptoms or signs and symptoms the patient had, and then if there's any treatment, if there's any medical history, this is a case report and this is a description. We also have cross-sectional studies where we're looking into different populations. We can have case series. It means that we have 10 case reports and then we combine them all, and then we are going to write a case series, I would say, report, because this is what we call a case series. It's coming from case reports. You can also look into the analytics to do the analysis. In this case, we are going to have a case control as a type. We can also have a cohort or observational type. So we observe, again, different populations, for example, or different groups. And we can also do what we call meta-analysis, meaning that we are going to have a type of sequential research where we look into the same population but 20 years ago in another country, but people having a specific disease, and populations now in our country having exactly the same disease but in a different population. But these are different times, these are different tools, different instruments, different sometimes results as well. But what we do is that we're going to use, again, meta-analysis, and we are going to perform statistical tests in order to compare the results that we obtained 20 years ago, for example, or 10 years ago as well, with the actual results that we have now. So we are going to write an article, or a report, based on the results from the observational analytic study related to, of course, meta-analysis. We can also have the type of the clinical trials, which is experimental. And here we talk about intervention. And the gold standard for the clinical trials is randomly assigned patients being, again, dispersed or grouped in different groups, with a control group where it can be a placebo or it can be a drug which is our competitor but it's a control, and our drug itself. So here we talk about randomized controlled trials. But don't forget something else, is that we have the randomized patients. We also have a control in our groups, but it also needs that we don't have any bias. This is why we talk about double masked or double blinded studies. It means that when we are giving the product or the clinical supply, neither the investigator nor the patient or the study subject know about what they are using. This is what we call double blinded or double masked. It's very interesting because in this case, investigators doesn't know if this is an experimental drug or the control, if that's a drug or a placebo, and the patient doesn't know neither. So we really remove, I would say, lots of bias from the results. So when we talk about randomized, controlled, double-blinded clinical trials, this is what we are talking about. It's for removing bias, and of course, this is an intervention that is again an experimental study, and this is a clinical trial. So now let's talk about the methodologies. What is the ideal clinical trial methodology? Clinical trials almost always should include a control group against which the new intervention can be compared to. Study participants should be assigned to control and intervention groups through randomization. Randomization means to assign by hazard someone in a group. So if you have 600 patients and we have 3 groups, we are going to give to each patient a number, patient number, and then we are going to add all of those numbers into an Excel file or a computer, which is a very simple, I would say, function to do. Excel also has a built-in function for that. And then we are going to ask the computer or the Excel to assign randomly those, 600 people into 3 groups based on the patient number. And then we are going to again have 3 different groups here, and now we may have one group having the I would say the drug A, another group drug B, okay, and the other group they are going to have the control, and the control may be the placebo, or the control may be an existing drug on the market. So here, when we talk about trials, here we talk about parallel trials. It means that we are going to have, patients in blue, in again dark blue, and in light blue. Each group is going to continue with the same treatment. This is the parallel groups. So patients usually they are screened, they are then randomized, distributed by hazard into groups, and then they are going to be exposed to the same, to a drug, to another treatment, different dose, or a placebo. And when we talk about randomized controls, randomized control studies are comparative studies with an intervention group and control group. All participants are equally likely to be assigned to either group. And again, here we also talk about a simple masking or double masking or simple blind or double blinded studies where the investigator and or the study subject may not know which, again, I would say product they are taking, or which dose, or if they are taking the placebo. So now let's talk about the efficacy and safety in clinical research. We talked about the pharmacodynamics. This is the biological effects of the drug on the body. So we talk about what the drug does to the body. So here we are going to use different doses, and then we are going to look into again the effect, if there is any. Even we are going to have establish some metrics looking into the efficacy profile of the drug. Sometimes it's dose dependent, sometimes it may not be dose dependent, depending on the drugs. And we also want to know about the mechanism, but there are many drugs that we don't know about the mechanism of action still, like antibiotics, because these are proteins, and these proteins, they are also again metabolized or degraded in the blood as well, but they do their work and then they are degraded and they become amino acids, and we don't know exactly again how they are working. Again, I'm not saying for all of the drugs, but for many drugs we don't know their mechanism of action, but we know their effect and about their efficacy. So here when we talk about pharmacodynamics, we talk about highest plasma, that dose concentration from same drug dose given to different subjects. We are looking into the amount of the drug available in the blood of patients. We think, or we talk about relationship between drug effects and plasma drug concentration, because you may have again higher level of the blood, of the drug in the blood, and then we may see some therapeutic effect. And then we may see less and less therapeutic effect. This is not again the case because we have to look into the volume of distribution of a drug. Some drugs, they stay in the blood. Some drugs, they also diffuse out and they go to the intercellular or interstitial fluid. Some of them, they go into the intracellular, they go in the cells. Some of them, they also go to different tissues like the fat tissue. Some drugs, they even go to the bone tissues. And then because of this, we will see that the washout period, so the time necessary for the body to get rid of the, obviously completely of a drug, depending on the nature of the drug, and of course its properties, it may be different. We have, for example, vitamin C that stays in your blood for only 1 hour. But we have warfarin that stays in your blood for 3 months. So here again, we look into the relationship between drug effects and plasma drug concentration, but we also need to pay attention and consider, of course, other physicochemical properties of the drug. We also look into the relationship between the drug effect and repeated dosing, because we may give 1 dose of the drug, we look into the effect. Of course, the pharmacodynamics, we may not have any effect, but we look at that. And then we are going to give several doses in order to reach a level that we call a steady state level, which is, again, the amount of the drug in the body that will going to maintain a certain concentration if we are following the same, following to the same dose administration with the, I would say, administration intervals or dose intervals. We also look into relationship of drug concentration in tissues versus plasma, as I told you, because each drug has a volume of distribution, and not all of the drugs are going to have the same pattern of being distributed in the body. Same thing applies to the same pattern for being metabolized. But here we are talking about pharmacodynamics, so about the metabolization, I'm going to talk about this in the next slide when I'm going to cover the pharmacokinetics. And then we also look into the relationship of drug concentration to drug effect at the receptor site. In each cell, we have receptors, we have effectors, we have intracellular receptors as well. We have channels, we have other, I would say, constituents of a cell as well. And the drug may affect directly or indirectly the functioning or the normal functioning of any, again, I would say, activities that a cell has. But again, this information comes from fundamental research. But again, there's a difference between in vitro studies and in vivo studies, because in vitro we can always isolate cells and look into the effect of the drug. But when we are giving a drug in a living organism, the cell may not be exposed as well as it was in vitro. And we have other factors like such as, again, different, I would say, feedbacks from other cells or neighboring cells to the target cells as well. So I'm not here trying to make it very complex, but I'm just talking about different factors that may affect the study that we do related to the pharmacodynamics. And then talking about pharmacokinetics, we always talk about the absorption, distribution, metabolism, and excretion of a compound. So we talk about what the body does to the drug. So pharmacokinetics helps determine inter and intra-individual variability in drug concentration and drug response among different individuals. So variability can be caused by drug-drug interactions, drug-disease interactions, pharmacogenetics, impaired kidney and or liver function, etc. That's why we can always see a type of a normal distribution when we look into the effect of a drug in a patient population. Meaning that when we give a drug to patients at different ages, we may, or ages for example, we may see that the drug is, for example, working better on again, the adult population. And here, when we are looking into a distribution or the frequency of the occurrences or the measurements that we did, we can see a bell-shaped curve and a normal distribution showing that the effect of the drug, again based on the age of the patients, is mainly concentrated in between, for example, age 25 and 45. And when we're looking into the pediatric population or the elderly population, we see that for some reasons, for a specific drug, I would say, we don't have that much effect. Again, it will depend on different drugs. But here I just use this example just to talk about the fact that we may have a normal distribution of the data when we are looking into the effect of the drug in a population. However, and you understand now very well that we have inter and intra individual variability in drug concentrations and drug response among different individuals. This means that if now a drug is causing some adverse drug reaction or side effect, in this case, we may not have a normal distribution of the data set. It means that, or frequencies. It means that we may see, for example, we have much more adverse drug reactions observed in the pediatric population and not in adults or not in the elderly population. In this case, we don't have a normal distribution. Why I'm talking about the normal and non-normal distribution? That's because we are dealing with clinical trials. We are measuring parameters. We chose variables and we are measuring the effect of the drug through different ways. So it may be looking into the blood glucose level. It may be looking into the size of the tumors or the diameter of the tumors. It may be just asking the patient about his or her quality of life. So this is another type of variable that we are looking at. And we may have different types of data. At the same time, we will see a different type of distribution of the data based on the age of the patients, for example. This is why I talked about the effect of the drug that can follow a normal distribution. However, we may not have the same pattern when we are looking into the adverse drug reactions, where we may, in just the case of a drug XYZ, we may see that, again, a specific population or a specific age, a population with a specific age, are going to experience adverse drug reactions. But this is not a normal distribution. And in this case, we are having a different type of distribution, hence different type of statistical analysis that we will need to use. We're going to look into this, of course, in more in depth in the next slides. So when we do the pharmacokinetic studies, of course, again, I'm showing this to you just to tell you that it's not going to be definitely or necessarily a company, I would say, data. Companies can decide to publish the information about the pharmacokinetics, even pharmacodynamics of the drug, because they would like also to establish scientifically, again, the effects of the drug in the medical field, or in the medical world. And this is why you will see several articles that are, again, explaining or establishing well the pharmacokinetic or pharmacodynamic profile of a drug, of an experimental drug. So now let's go over the design of the studies. We already talked about the parallel design, where we have here individual A and individual B, where each of them is going to receive a treatment and they will continue till the end. And then we finish the last visit, last patient, last visit, and then we are going to look into cleaning the data, closing or freezing the database, and then to do the statistical analysis. But we also have another design of the study that we call crossover. So on the top left side in the table, you can see that you can have individual A that is going to have a treatment, and then we are going to stop the treatment, and we are going to put individual A, the treatment A, and we are going to put the same individual now in treatment B. So one individual here had treatment A, then after the washout period had the treatment B. So here we have an individual that served as his or her own control. And then we also have a treatment B, as you see, where one individual was in treatment B and then after the washout period had treatment A. So here, this is a design in the study that we call crossover, where we are having individuals who are going to be exposed not only to the active drug, but also to another group or another dose of the drug, or maybe the control. So each individual is going to serve as his or her own control. It's a very, very interesting concept. And again, this is as opposed to, again, the parallel design. So now on the right side, what you can see is all of the, I would say, all of the points that we have to consider when we are doing clinical trials. So for example, when we are going to do clinical trials, we need to also look into the route of administration, because it's obvious that the dose that we are giving, if this is an ingestion, the drug that is absorbed by ingestion is different from the dose when this is by, again, injection. Okay, so we have to pay attention to that as well. We also look into the duration of the drug exposure. It means that we are going to give a dose, perfect, like you can see on the top in the middle of the right, quadrant here. You see that we have a single dose or steady state. With a single dose, we give a drug and we are going to look into the effects. But we may now, before the effect ends, to give another dose. And then before the effect of the second dose ends, we give another dose. So we continue to give doses based on a dosing interval. And we are going to see a multiple dose, I would say, administration, where we will reach a level where we have a type of steady state, or I would say a level of drug that is going to be within the therapeutic window, and it's going to continue to be plus minus 10-20% the same concentration in plasma. So we also look into the single dose or steady states. We also look into the sampling strategy. What we are going to sample. Are we going to take urine samples? We're going to take saliva samples. Are we going to do any biopsies? We have to look into what are the samples that we have to take in order to test the efficacy and the safety of our drug. We also look into blinding, because we want to remove the bias. So we need to also look into, hiding the drug or the placebo, so that investigators or patients are not going to have the feeling that they are, taking a drug or they are taking the placebo. So in this case, if they don't know, they are not going to have any factor that may affect their psychology. And even their feeling about the drug that they are testing. And it's going, again, as I told you, removing the bias. We also look into the pharmacodynamics endpoints, and we may also look into the influence of genes on the extent of the action of the drug. Here we talk about pharmacogenetics or genomics. And so in a nutshell, this is just for talking about the study design for this slide. Now, coming back to the statistics, as I told you, we are going to have numbers. We are going to have qualitative data as well as quantitative data. Now, when we are going to have, I would say, millions of data coming from a study, we need to find ways to not only to present the data, but also to analyze the data, because we have our outcomes, primary, secondary, tertiary, quaternary outcomes that we would like to test with statistics. However, if we have only qualitative data, we will not be able to do inferential statistics. We will need to do what we call descriptive statistics. If we have quantitative data, so having numbers, here we can, of course, do what we call inferential statistics. So let's go over what is a descriptive statistics. What it does is that it summarizes the demographics of the study population, such as age, gender, ethnicity, disease, etc. And descriptive statistics measure the distribution central tendency, and this is where we talk about the mean or median, and variability or dispersion. Here we talk about range, quartiles, coefficient of variance, or standard deviations. Here we call that SD. We can also talk about the standard error. So these are all different measures that we do when we are dealing with again data that again is going to allow us to perform what we call descriptive statistics. So coming back to standard deviation, a standard deviation or SD here is different than the standard error of the mean that we write as SEM. So SEM or standard error of mean quantifies uncertainty in estimate of the mean, whereas standard deviation indicates dispersion of the data. So SD is the standard deviation describing the variability of a sample size. And SEM is again a standard error that doesn't describe the sample but describes the uncertainty of how the sample mean represents the population mean. If you would like to calculate the standard deviation or the standard error or the range or quartile or coefficient of variance or the mean or the median, there are different formula that you can use, of course. But here I would like to talk about, again, not the formula, because this is, again, you can do this in databases. You have the SAS, SPSS, even with Excel, built-in functions. It's very useful. You also have R Studio, for example. You have so many of those statistical softwares that will allow you to calculate all of these different, I would say, measurements. But talking about why or how we are going to use these different measurements, again, is that in descriptive statistics, we summarize the demographics of the study population. And in order to look at that, I'm going to go over, again, the descriptive statistics in the next slide. So here, you can see in table 1 the demographic and clinical characteristics. So we talk about number of subjects, the gender, male number, percentage, okay, age, years, body weight in kilogram, body mass index, for example, concomitant medication, and biochemical parameters. So as you see, there is no analysis. We are just giving demographic and clinical characteristics. Now, when we talk about the age, as you can see in under group A, we give the age, but we put plus or minus. So this is the average of the age plus or minus the standard deviation or SD. For the body weight, we can say we have 200 patients. Their average body weight is 75 kilograms plus or minus 10. The standard deviation. For the body mass index, I would say the average of the body mass index is 26, 27. Okay, maybe obese one, so 29, and then plus or minus 1 or 2. The standard deviation. So this is the way that we are going to show demographics and clinical characteristics in a study coming from the data that we obtained from our patients, and this is related to descriptive statistics. Now, if we would like to look into inferential statistics, this is when we are dealing with quantitative data. So inferential statistics uses different statistical tools to make conclusions about a population by examining random samples. So we can also use geometric mean ratio and 95% confidence interval, again, in drug exposure testing. I'm sure that you heard about student t-test and analysis of variance, ANOVA, some of you, of course, ANCOVA as well. You heard about several statistical tests, but what I'm going to tell you now is not only about, again, t-tests, it's mainly talking about different statistical tests that we use, but based on the distribution of the data. Because if we have a normal distribution of the data, the tests that we are using are different from when we have non-normal distributions where the distribution is skewed. So coming back to here, I'm going to again review these. One of the notions that you need to know is about the paired and unpaired data. Paired and unpaired data is when you're having, again, people, these are all, again, dependent samples, so we are dealing with the same individual at different time. We talk about the paired, data. But if we are not dealing with the same individuals at different times, we call that unpaired data. We also have dependent samples and independent samples. What is that exactly? Depending whether or not you are using completely separate and different populations in different groups, or if you are using the same population but with another dose of a drug, another dose of the drug, but they're all the same population, we have different definitions of the samples. Hence, different statistical tests that we use. So if we are having independent samples, that's what you can see on the left lower part of the slide, we are having three groups here, and each group is independent from another group. We have, for example, we have a drug, we have a placebo, we give the placebo to group 1. And then we have a drug at 2 milligrams per kilogram, for example, we give it to group 2 in the middle. And we give the same drug at 6 milligrams per kilogram, we give it to the last group that you see here. So these are three distinct groups, and these are independent samples. So since they are independent, the test that we use is going to be a different test, and that we're going to cover, of course, in the next slides. However, if we are looking into the dependent samples, here we are talking about the same groups, the same individuals. We have individual 1 on the top, and under the dependent sample line or column, we see the first individual, and then we see some intervention after some time. And we are now going to look into, again, another intervention or the same intervention but continuation of the dose in the same individual. This is like you are having, again, a group of individuals and you're asking them to run, okay, and then to come and you're going to measure their heart rate, okay. In a study, we measured the heart rate of a group of 6 people before running and after running. Here we have the same group, before and after intervention. Here we are dealing with a dependent sample. But if we had 6 people and we were looking and we measured their heart rate, and then we asked 6 other people, different people, asking them to run, and then we would get their heart rate, we can still do the statistics, but here our samples is independent. So we will need to use a statistical test for independent sample. Now you understand the differences. We also have dichotomous variables. These are variables that's a type of categorical variable that has only 2 distinct categories or levels. That's why this is called dichotomous. So gender, for example, in clinical trials, we have male and female, we have pass and fail, we have alive or dead, true or false, yes or no, present or absent. These are all dichotomous variables. So this is yes or no, I would say, works or not, doesn't work. So this is, again, another term that you need to know when we talk about inferential statistics in clinical trials. Now let's go over the distribution and tests. On the top left side of the slide, you can see a normal distribution. When you look at the y and x axis, you're able to see the number of individuals, based on the response to the same dose of a drug. So we gave a drug, the same dose of a drug, to several individuals, and we looked into the response to the same dose, and we looked into resistant individuals that have minimal effect. We looked into the average effect of the drug, and we had the majority of individuals, and we also had the maximal effect where we had sensitive individuals. So here we see that the majority of individuals, following of course the bell-shaped curve for the distribution of the responses, is normal. So this is a normal distribution. We looked into the effect of the drug response to the same dose. On the right side of the slide, top right side, you can see the number, so the counts of the adverse drug reactions or side effects observed in patients at different ages. So we gave them the same drug, and we observed that the majority of adverse drug reactions are experienced by individuals in between age 20, I would say 19, 21 and 30, 33. But we see that the overall tendency is that we do not have as many side effects, adverse drug reactions. We don't observe them if individuals are aged more than 33, 34. So here you compare this distribution with the previous distribution on the left side of the slide. You see that the left side has a normal distribution, because if you look at the average or in the middle point, you see that you have almost the same population on the left or right side of the average point or the central point. But on the right side, on the right graph, we see that the distribution is not normal. This is what we call a skewed distribution. And in this case, since the distribution is not the same, we don't use the same statistical tests or inferential tests. So allow me now to recapitulate for the slide. Look into the slide in the picture in the middle, or I'll say bottom middle. Here you have the on the left the data normally distributed, and in the other one you see that the data is not normally distributed, so it's a non-normal distribution. This is a skewed distribution. In case of a normal distribution, we use tests that we call parametric tests. So normal distributions, we look at the answers, the frequencies, and if we have a bell-shaped curve, we see that this is a normal distribution. Hence, we need to use tests that we call parametric tests in order to test our hypothesis. And the examples of those tests are student t-test, analysis of variance or ANOVA, or the Pearson correlation. But if the data is not normally distributed, then we use non-parametric tests. And here we can talk about tests like Mann-Whitney U-test or Spearman test or Wilcoxon test. So it's very important, because the distribution of the data is important. And if you have a normal distribution, you can make assumption based on your findings. If you have a skewed distribution, you can always do non-parametric tests, but you cannot make assumptions. Okay, these are just observations, and this is again the fundamental difference in between normal distribution with parametric tests and non-normal distributions with non-parametric tests. Let me explain it in a more simple way, because now you understood again what is a distribution which is normal or abnormal. Now I'm going to make it more simple for you to understand. So here on the top left side we have the data which is normally distributed, and on the right side we see that the data is not normally distributed. We look at our data, if you have a bell-shaped curve or not. Now you are having millions of data, and you're now looking into data sets. You also created your graph with the frequency, and then you saw that your data is normally distributed. For example, the heart rate after after taking a drug. And then you will now you want to test the heart rate of your patients after taking a drug with the heart rate of the same patients after taking placebo. So you're going to have two columns, both of them heart rates, before intervention, after intervention, and we know that they are coming from the same population. So this is, again, these are dependent samples. Now, what tests you are going to use? You are going to see that if you have data normally distributed, you need to use parametric tests. These are all of the tests in the section of this flowchart, which is in a dark blue or blue on the left. So you can see the parametric tests. For parametric tests, if you have one sample, we use a t-test or z-test, depending on the number of samples. And if we have two samples, in this case, if we have independent samples, we're going to do a test, z-test or t-test for independent samples. And if not, if we have paired samples, we're going to use the paired t-test. So depending on your distribution of your data, if this is a normal distribution, you can only use parametric tests in order to test a hypothesis. So here I talk to you about the heart rate of patients, of one patient population, but with placebo or without, or with a drug and with a placebo. But this is the same sample, so this is a dependent sample. So we are going to use a t-test for dependent samples. Now let's go over the adverse drug reactions. We know that in the example that I gave you, we have a skewed distribution. So we have mainly patients between the age of 19 to 33 who are going to have an adverse drug reaction. But we need to compare that. We know that the distribution is not normal, but we still have a hypothesis to test. We still want to know whether or not patients are going to get more adverse drug reactions when they take drug compared to if they are taking placebo. So we have again two groups, placebo groups exposed to the drug group, and we are looking into adverse drug reactions experienced by patients at several ages. And we would like to test whether or not the drug did cause the adverse drug reactions. Here we look at the distribution of the data, and we see that this is not, as you see on the top right side, it was not a normal distribution. Hence, we have to use non-parametric tests. So here, if we have one sample, we are going to use the test chi-square or KS, or runs test or binomial tests. If we have two samples, in this case, we are going to use independent sample or paired sample. This is where, again, if we are dealing with the same population for the adverse drug reactions, these are, again, dependent samples. So we are going to have, again, the paired sample. So we are going to use the sign test, Wilcoxon, McNemar, or chi-square test in order to compare the population before the, without the drug, placebo, and after the exposure to a drug for the occurrence of adverse drug reactions. So you understood now that in clinical trials we have variables. We write the variables, of course, in a protocol. We also write about the statistical analysis that we are going to use. We are also going now to expose patients to different interventions or no intervention like placebo. And then we are going to get the data. We need to express or give some demographics about the data, so we are going to use descriptive statistics, as I already showed you a table. And then we would like to do inferential statistics, so we want to compare I would say two groups in order to test the effect of a drug. So to one group to the same group we gave the placebo, and then to the same group after 2 days, 1 week, we give the drug, and we are going to measure the same variables like their heart rate. Since this is the same group, this is a dependent sample, and since we are having a normal distribution, we are going to use a t-test for dependent samples. But now if I would like to look into the occurrence of adverse drug reactions or side effects in the same group as well, I'm going to have the same group before I would say being exposed to placebo, and I'm going to look into all of the adverse drug reactions that they may tell me, and I'm going to write them. I get the data from there, and then I'm going to in 1 week I'm going to expose the same group to a drug, experimental drug, and then I'm going to again collect information about the adverse drug reactions. And now I'm having adverse drug reactions for the same group, so this is a dependent sample or paired sample, and this is before and after intervention. Here, since I have a non-normal distribution, I will need to use non-parametric tests. And in this case, I'm going to use a non-parametric test for two samples, paired sample. Now I can compare using a non-parametric statistical test to compare the occurrence of adverse drug reactions if patients are exposed to again a placebo or to a drug using a test like chi-square, McNemar, Wilcoxon, or Syme. So this was, in a nutshell, all of the statistical concepts for clinical trials. Okay, so now some words about the parallel groups. We already talked about those. I also explained to you this parametric and non-parametric tests, and you understood very well. So now I'm just going to read the slides, the next two slides, just to recapitulate what I just explained to you in the last two slides. So parallel group design compares the outcome of an untreated group to the outcome of the treated group. So with an intervention versus drug or drug. Different statistical analyses can be carried out. We call them parametric tests. So a student t-test for paired data and two independent samples when the outcome follows a normal distribution. We may use a Fisher's exact test to compare two proper proportion when the outcomes is a binary variable, for example, which with improvement or no improvement, dichotomous, yes or no. And ANOVA compares more than two groups that follows a normal distribution. So when we have a normal distribution, if we have to compare two groups, we use student t-test. If we want to compare 3 and more groups, we are going to use ANOVA. So this is why we talk about t-test and ANOVA. They are both parametric tests, and they are again applicable to normal distribution. Now let's talk about non-parametric tests. So different statistical analyses can be carried out. Again, these are, we call them non-parametric tests. So I'm going to cite them. A Wilcoxon rank sum test or a Mann-Whitney test can be used to compare two groups and when the outcome follows a non-normal distribution. So here we talk about extreme outliers that are present. So if you have a non-normal distribution and you have a skewed distribution and you would like to compare two groups in your clinical trial, one group exposed to placebo and the same group being exposed to the drug after a week, you have to use a test like Wilcoxon rank sum test or Mann-Whitney test. These are non-parametric tests for non-normal distributions. Now, a Wilcoxon signed rank test is used for paired data and when the outcome follows a non-normal distribution. That's what I mentioned. Now you can also have a Kruskal-Wallis test. This is to compare more than two groups that follows a non-normal distribution. Of course, extreme outliers present. So as you see, as opposed to parametric tests where we had t-test and we had ANOVA, t-test was for two groups, ANOVA for three groups. Here, in the non-parametric tests for non-normal distribution, we have Wilcoxon rank sum test or Mann-Whitney test for two groups, and we have Kruskal-Wallis test to compare more than two groups. Perfect. So I hope that you enjoyed today's presentation. I know that it was complex, but it was complete. And again, we covered today all of the, I would say, the statistical that we need in clinical trials. So thank you so very much for your time. I wish you all an excellent day. Take care.